Hi everyone, welcome to our video tutorials, and today we're going to talk about how to use a protein sequence database called PFam. Now, as I said, PFam is a protein sequence database, and PFam organizes proteins based on sequence similarity. So first we want to go to PFam, and we can just go to Google, type in PFam, and I'm going to take the first hit, pfam.xfam.org. From the PFam homepage, I find the easiest way to find what I'm looking for is to go up to the search tab at the top. You can browse and search for specific things if you know, for example, the type of protein that you're looking for. You can type in the jump to, or you can type in the keyword search. I find those a little bit less reliable and not really useful for what we're going to do. So again, we want to go with the search tab at the top. So when you get to the search page, you'll notice that it's asking you for the protein sequence that you're interested in. So in order to get that, the most efficient way, I think, is to go back to the protein data bank, and I'm going to go with my protein 4R78. From here, like I said, what I really want is the protein sequence. So if I click on this link, display file, and then FASTA sequence, I'm given the amino acid sequence of my protein, 4R78. So if I copy my protein sequence, go back to PFam, paste it in the search box, and press submit. This can take anywhere from seconds to minutes, so be patient. If it takes more than 5 minutes, I think you should maybe start over again. Okay, so in this case it took about 15 or 20 seconds. That's not too bad. So when PFam shows you the results, the first thing that we notice in the middle of the screen is that PFam has identified our protein as a choline kinase. We already knew that, so it's good that it found what we expected it to find. The next thing that you want to look at is where it says significant PFam A matches. Everything in this section is something that you should pay attention to and look at. So in our case, we only got one match. Choline kinase family. We knew that again. So if you start scrolling across the screen, you'll see something called HMM length. The HMM is hidden Markov model, and it's a statistical model. It wasn't designed for protein sequences, but it works really well. So the HMM, or the hidden Markov model, is the defining characteristic, or the fingerprint, if you will, of your protein. So PFam has this fingerprint in its database, and it's going to compare the sequence that you search against the HMM. In this case, the HMM is 213 amino acids long. And if we look at our alignment, we can see that we are actually aligning from residue 2 to residue 210. So that's a fairly significant, sizable alignment, which is great. The next thing that you should be interested in is the E value, or the expect value. This is similar to what you'll find in a blast search. And that means that E values that are very small, in our case 10 to the minus 37, that's a great hit. So the smaller the E value is, the better the alignment quality is. If you're curious, you can show the alignment and have a look. In the top row, you'll see the HMM residue definition. In the bottom row, you'll see your amino acid sequence that you searched, and it's color-coded. So these color codes tell you how well it matches the HMM definition for your family. Green means 100%, red means 0%. So in our match, you can see that almost all the residues are green, which means a very high quality hit. So I'm going to click on the choline kinase link, and then I'm presented with a Wikipedia article that tells me all about the choline kinase family. You'll also notice along the top is a navigation bar, and along the side is another navigation bar. These two are nearly identical. So clicking on sequences here is the same as clicking on alignments on the left-hand side. So if I click on alignments or sequences, I can see that this family consists of 3,480 protein sequences. If I want to look at the alignments, I can do that by clicking on any of these links. So for example, the HTML view of the seed 33 proteins. Here they are. I can scroll down through them and see how well they align with each other. I don't recommend clicking on the HTML view for the full 3,480 sequences. It probably will crash your browser. I know from personal experience. If I go back to the summary page, another thing that I can look at is the species distribution. So here I'm presented with a nice graph that tells me roughly 25% of the proteins in this family are found in bacteria. The other 75% are found in eukaryotes. And I can trace this down. Here are mammals, primates, humans. So there are 20 sequences of choline kinases that are present in humans. Again, if I go back to the summary page, another useful feature is to look at the number of structures. So if I click here, I can see that 39 protein structures or three-dimensional structures have been determined for this family alone. And if I click on the individual structures, I get information specific to those proteins. In this case, I can click on and I can see which ligands are present in this PDB entry. So here we have a sulfate in our protein structure. Sulfate is usually a crystallization artifact. It's not very interesting for us. It's not a substrate that we're interested in for catalytic studies. So I think that's the most important stuff that you can learn from the PFam database. Thank you.